Welcome back to the Deep Dive. Today, we're going to be diving into a recent research letter from JAMA.
Okay.
It's titled Antibody Response to Respiratory Syncytial Virus Vaccination in Immunocompromised Persons. It was published right at the end of December.
Okay.
And I know you specialize in transplant and oncology infectious diseases.
Right.
So this is right up your alley, I think.
Absolutely.
They looked at how well the new RSV vaccines are working in immunocompromised patients.
Yeah.
Which is something that, we haven't had a lot of data on.
That's right. Yeah, since these vaccines were approved,
Right.
we've all been kind of trying to figure out how best to use them in this population.
Mm.
We already know that RSV can be a serious problem for immunocompromised individuals.
Right.
But the initial trials really focused on healthy adults.
Yeah.
So this research is really helpful to help fill in some of those gaps.
Exactly. And it's so important because, RSV in transplant oncology patients can lead to some pretty serious complications.
Yeah.
Pneumonia, respiratory failure, even death.
Absolutely.
It's not something we can take lightly.
Yeah, the new pre-FRSV vaccines, both the adjuvanted RSV pre-F3 AS01E
Right.
and the non-adjuvanted RSV pre-PF, have been great at preventing severe disease in the general population.
Right.
But we need to know, do they work as well in our immunocompromised patients?
Exactly.
And that's really where the study comes in.
Yeah.
It looks specifically at antibody responses in this vulnerable group.
Okay, so let's get into the specifics of this study. How did they go about answering this question?
Well, they used data from a national cohort study that was already looking at viral vaccination and infections in immunocompromised individuals.
Mm-hmm.
This included patients with a range of conditions,
Right.
Things like solid organ transplants, rheumatoid arthritis, and lupus.
Okay.
The patients in the study chose to receive either the adjuvanted RSVA AS01E vaccine
Right.
or the non-adjuvanted RSV AB vaccine, which is a good reflection of real-world practice.
So it's, a diverse group of patients getting different vaccines, just like what we see in our clinics.
Exactly.
Did they follow these patients over time?
They did, yes. They collected blood samples from the participants
Okay.
at several points.
What exactly were they looking for in these blood samples?
They were primarily interested in two key indicators of immune response.
Okay.
First, they measured the fold rise in pre-FIgG antibody levels from baseline to 4 weeks post-vaccination,
Okay.
with a 4-fold or greater increase being considered seroconversion.
Right.
This basically tells us how well the body is responding to the vaccine by producing antibodies.
Okay, so that gives us a sense of the quantity of the response.
Right.
What about the quality of those antibodies?
That's where the second measure comes in.
Okay.
They also measured the neutralizing antibody activity, or NT50, at 4 weeks.
Okay.
This tells us how effective the antibodies are at actually blocking the virus from infecting cells.
So they were looking at both the amount and the potency of the antibody response.
Right.
It makes sense to look at, both sides of the coin there.
Right.
You mentioned a high-titer neutralization threshold.
Yeah.
How did they determine that?
They compared the neutralizing antibody levels in their vaccinated participants
Okay.
to a high-titer reference serum collected from individuals before the RSV vaccines were available.
Got it.
This gave them a benchmark to assess how strong the vaccine-induced immune response was.
Interesting. So it's like having a historical control group to see how the current vaccines measure up.
Right.
Okay, I think we've got a good handle on how the study was designed.
Yeah.
Let's move on to what they actually found.
Okay.
I'm ready. What did they discover?
Well, of the 139 participants who received the RSV vaccine,
Okay.
38 had both baseline and 4-week post-vaccination samples.
Okay.
Most of the other participants were excluded simply because they didn't have that initial baseline sample.
Okay, so we have a smaller group to look at for the results.
Right.
Now, remember, the big question we're trying to answer is, how well do these vaccines work in immunocompromised people?
Right.
Do they have a similar response to the general population?
Well, that's what they were hoping to figure out. And
Right.
the first key finding was that the antibody response was actually quite varied in this immunocompromised group.
Okay.
The median pre-FIgG fold rise was 4.21.
Mm-hmm.
But only 61% of the participants achieved seroconversion by 4 weeks.
That's a notable difference compared to the almost universal seroconversion we typically see in immunocompetent individuals after vaccination.
Right.
What about the participant characteristics?
Yeah.
Did they find any trends in those who did and didn't seroconvert?
Interestingly, the characteristics of the participants who seroconverted and those who didn't were very similar.
Huh.
This suggests that other factors beyond their specific condition or medications
Okay.
might be influencing the immune response.
That's definitely something to think about. What about the neutralizing antibodies? What did they find there?
When it came to neutralizing antibodies, 58% of the participants reached that high-titer neutralization threshold at 4 weeks.
So again, not everyone developed the potent neutralizing antibodies we would hope for.
Exactly.
This variation in antibody response is, a key finding, especially compared to the more consistent,
Right.
robust responses that we see in the general population.
Right.
This definitely raises questions about how well these vaccines might protect our immunocompromised patients.
Yeah.
Are there any other details
Yeah.
that might help us understand these findings?
There are actually a couple of really intriguing details that emerged.
Okay.
Details that might offer clues about how to improve vaccination strategies in this group.
I'm all ears. Tell me more.
Okay. One of the most surprising findings was that individuals taking mTOR inhibitors were actually more likely to achieve high-titer neutralization compared to those who weren't on these medications.
That's unexpected. We don't usually think of mTOR inhibitors as, like, boosting the immune response.
Right.
Any ideas why that might be the case?
Honestly, the researchers didn't dive too deep into the reasons behind this finding.
Okay.
And we have to remember this was a fairly small study.
Right.
But it does raise some really fascinating questions about how mTOR inhibitors might be affecting the immune response to vaccination.
Yeah, it makes you wonder if this could change how we approach vaccination in transplant patients who are on mTOR inhibitors.
Yeah.
We definitely need more research to confirm this finding and, figure out what's going on.
Right.
But it's something to keep in mind.
Yeah, I agree.
Okay.
And there was another interesting observation that could be important for vaccination strategies going forward.
This is fascinating. What else did they find?
The data suggested that the adjuvanted vaccine, RSV-A AKO1E, might be leading to better neutralization than the non-adjuvanted RSV-AD vaccine.
That's interesting, but it makes sense.
Yeah.
We often see a better immune response with adjuvanted vaccines, especially in people who are immunocompromised.
Exactly. Adjuvants are designed to give the immune system an extra boost.
Right.
And this study suggests that this strategy could be particularly beneficial for immunocompromised individuals getting the RSV vaccine.
So if we take a step back and look at all the results together, what's the main takeaway for clinicians like us who are working with immunocompromised patients?
I think the key message here is that while these new RSV vaccines are a great tool for preventing severe RSV, we can't assume that a single dose will be enough to protect all of our immunocompromised patients.
Yeah, you're right. We can't just extrapolate from the data on healthy adults and expect the same response in this population.
Absolutely. And this really highlights the need for more research to figure out the most effective vaccination strategies for this group.
Right.
Should we be considering additional doses, booster shots, or even completely different types of vaccines?
Right.
These are all important questions that this study brings to light.
And finding those answers could have a significant impact on the health of our patients,
For sure.
especially as we head into RSV season.
Exactly. This research is a good starting point, but we need more information to guide our decisions and make sure we are doing everything we can to protect our immunocompromised patients.
Okay, so we've talked about the methods and the results of the study.
Right.
Now, what are the broader implications of these findings?
Yeah.
What does this research mean for the field as a whole? This research letter really does offer some valuable real-world data on the antibody response to RSV vaccines in immunocompromised individuals,
Yeah.
which we know has been lacking.
Yeah, I think one of the most important takeaways is that a single dose of either vaccine might not be enough to generate a strong antibody response in a significant portion of this population.
Yeah, this difference in seroconversion rates, really has me thinking.
Yeah.
In my own practice, I've had a few patients who, didn't seem to mount as robust of a response as I would have expected. What factors could be contributing to this variability?
Well, it's likely a complex interplay of factors.
Okay.
The type and severity of the immunocompromising condition,
Right.
the specific immunosuppressive medications they're on, their overall health status,
Mm-hmm.
even things like age and prior exposure to RSV could all play a role.
That makes sense. It's not a one-size-fits-all situation.
Right.
So what does this mean for how we approach vaccination strategies moving forward?
It really underscores the need for more tailored approaches.
Okay.
We need to consider things like additional doses, booster shots, or perhaps even different types of vaccines altogether.
While adjuvants are generally known to enhance immune response,
Yeah.
are there any concerns or potential side effects we need to be aware of when considering adjuvanted vaccines for this specific population?
That's a great question.
Yeah.
While adjuvants are generally safe,
Right.
they can sometimes lead to increased local reactions at the injection site,
Okay.
like pain, redness, and swelling. There's also a theoretical concern that they could potentially trigger or worsen autoimmune conditions.
Right.
However, the data on this is still limited,
Okay.
and the potential benefits of enhanced protection likely outweigh the risks in many cases.
That's a good point. The potential benefits versus risks are definitely something we need to consider carefully for each patient.
Absolutely.
It seems like we need more research to figure out the optimal way to vaccinate this vulnerable group.
Yeah.
What other questions do you think future research should address?
Well, one of the most pressing questions is how these antibody levels actually translate into protection in the real world.
Right.
Just because someone doesn't develop a robust antibody response
Yeah.
doesn't necessarily mean they're going to get sick.
Right. There are other parts of the immune system, like
Right.
T cells,
Exactly.
that contribute to fighting off infection.
Exactly. So we need to understand the interplay between antibodies and other immune components.
Right.
Do those who achieve seroconversion experience fewer RSV infections?
Right.
Are their illnesses less severe?
Mm-hmm.
Answering these questions will be critical for determining the true impact of these vaccines.
This deep dive has definitely given me a lot to think about as I approach RSV vaccination in my own patients.
Good.
What are your final thoughts for our listeners,
Sure.
particularly those working with transplants and oncology patients?
I think it's important to remember that we can't rely solely on the data from healthy adults when making decisions about vaccinating immunocompromised individuals.
Mm-hmm.
This research highlights the need for a more individualized approach.
Yeah.
We have to consider each patient's unique circumstances,
Right.
weigh the potential benefits and risks,
Yeah.
and stay informed about the latest research to ensure we're providing the best possible care.
Absolutely. Knowledge is power.
It is.
And this study certainly arms us with more information to navigate these complex decisions.
Yeah.
Thanks for taking the time to break this down with us.
My pleasure. It's been a great discussion.
And to all our listeners out there, thanks for joining us on the Deep Dive.